The next section of the Cellular Level of Organization chapter concentrates on protein synthesis. Protein synthesis is a crucial aspect of how any cell works. It is how our genetic information directs the cell to make proteins, and proteins are the in-the-trenches workers for the cell. They do pretty much everything. They're support structures in the cytoskeleton. They're enzymes to run chemical reactions in the cell. They're signaling molecules. They're receptors to signaling molecules. There's just so many things that proteins do. They're just crucial. Understanding protein synthesis is an important part of understanding the physiology of the body, because proteins are at the base of almost every physiological process. This image illustrates the central dogma of molecular biology, which is essentially how protein synthesis takes place. The genetic information in the DNA, kept in the nucleus, is transcribed or copied into RNA, specifically messenger RNA, which leaves the nucleus and goes into the cytoplasm, where the ribosomes read it and translate it into protein. This is another representation of that central dogma of molecular biology. The DNA, which is double-stranded, is made up of a sequence of nucleotides, each with its own nitrogenous base, represented here by the letters A, T, C, and G. Those bases bind to particular other bases, A to T and C to G and vice versa, so that one strand has a particular code to it and the opposite strand has the mirror code or complementary code. The template strand is a particular side of that, and it's transcribed by RNA being built from it. And RNA is made up of the nucleotides with bases A, U, G, and C. A in RNA will pair up with T in DNA. U in RNA will pair up with A in DNA. And then C and G are the same between RNA and DNA also. And so the transcribed RNA, or messenger RNA at that point, moves into the cytoplasm, where the ribosome translates it into the protein. DNA and RNA have a very, very similar language, code, structure, however you want to describe it, whereas proteins are made up of chains of amino acids. And so in translation, the ribosomes are taking the nucleic acid code and changing it into the amino acid code. 3 nucleotides in the genetic code translate to a particular amino acid. With the 3 amino acid code, what's called a codon, it's possible to come up with 64 combinations of those. And that's plenty to code for the specific 20 amino acids that are available. And as you can see here, the AUG code in RNA, which is based on the TAC code back in the template strand, stands specifically for the amino acid methionine, abbreviated MET. The DNA GGC, represented in RNA as CCG, the complementary codon, stands for proline, etc., etc. And so a specific order of amino acids is built based on the code that's originally in the RNA. The DNA is just instructions for how to put together a particular sequence of amino acids there. This image illustrates the state of DNA, so to speak, in the nucleus. It's in chromosome format. The X-shaped chromosome here is actually slightly misleading. The chromosomes are in the more spaghetti mash looking configuration that you usually see in nuclei that aren't dividing. However, as we get down to the chromatin and the coiling structure we see with the histones, that's representative of what DNA is going to look like. And looking at the DNA off of the histone, there's the double helix structure, and if you can unwind the DNA and look at the two strands to each other, you can see the complementation of the nucleotide sequence. A and T always go together, and G and C always go together, and so the one strand is going to be the complement of the other strand, and one of those, depending on particular genes, is going to be the template that has the exact code of how to put amino acids together. The process of transcription is directed by a enzyme called RNA polymerase. The ASE ending is just referring to it being an enzyme, and then the rest of the name tells you what it does. It makes a polymer of RNA. What it does is it reads the template strand, finding an A in the DNA and pairing that with a U for RNA, a T in DNA paired with an A in RNA, a G in DNA paired with a C in RNA, etc., etc. And it connects a nucleotide onto that chain one after the other based on which one pairs up with the complementary template strand. The transcription of RNA from DNA is controlled by other regions of DNA called the promoter. They'll be upstream or in front of the gene usually, not necessarily immediately. Sometimes there's a little bit of space there. But transcription factors are other proteins that recognize the promoter, and when they bind to the promoter, they promote the binding of RNA polymerase to the gene so that it can transcribe the gene. Which genes are used in a particular cell is based on this mechanism, and that's what makes a muscle cell different from a skin cell and different from a neuron and all of that sort of thing. But it tightly regulates which genes are made and when they're made. Oftentimes the transcription factors are available based on environmental cues or changes in the body at a particular time. The gene in the DNA actually contains some sequences that aren't coded into the protein eventually. Those are called introns, and the other regions called exons are the actual code for the protein. There's a step in the movement of RNA out of the cell from when transcription is done before translation begins, where the introns, the part of the gene that isn't necessary, is removed, and the exons are then spliced together. Exactly what introns are for is a bit under debate, but one simple explanation might be that it helps to protect the genes. Mutations can occur in DNA at a given rate, and if the introns are there, they sort of decrease the likelihood that an important part of a protein is going to be mutated. There are other reasons. They might have some part to play in regulating when genes are expressed, and there are some genes that are rather complex, and they can rearrange the exons so that different proteins can be made from the same gene depending on conditions in the body. The process of translation is carried out by the ribosome. There are two parts to a ribosome, a large and a small subunit. When the messenger RNA moves out into the cytoplasm, these two subunits attach to the RNA. They find specifically the first codon, the first three-letter sequence within the code, which is always the codon for methionine, a particular amino acid. And in the large subunit region, the building of the protein takes place as a different type of RNA called tRNA, or transfer RNA, comes in and recognizes the codon. So it is complementary to the three parts of the codon. So AUG complements UAC, and the tRNA binds in the right place. It has the amino acid attached to it. The next tRNA comes in and recognizes the next codon, bringing its amino acid in, and the ribosome then takes the amino acid off of one tRNA and attaches it to the next amino acid. And then another tRNA comes in with its amino acid, and the ribosome grows the amino acid chain that way. And that just happens over and over again, reading the codons, until a special codon called a stop codon, one that does not code for an amino acid, is reached. No tRNA can come in bringing another amino acid, and so the chain just ends at that point. And that's all done within the ribosome that's attached to everything. This image comes from the chemical level of organization chapter, and it shows the levels of structural organization of proteins. The primary structure, the chain of amino acids represented at the top, is the result of translation, just connecting amino acids together by peptide bonds. Once that happens, then the protein folds, and so the secondary and tertiary and quaternary structure, if it's present, comes into effect as the protein folds. Protein folding is rather complex, and there's more to be said about it, but we make proteins that way. And the different ways that proteins can fold based on the specific amino acid sequence gives rise to an amazing number of types of protein. This image is from a common cell biology textbook that's used in upper level biology. And it shows a variety of different types of proteins and their shapes. From the very fibrous collagen protein, which is a supporting structural protein in many tissues, to the very specific shape of the porin proteins, which are channels for water or those sorts of things. There are specific proteins that can bind to DNA, as is shown in the deoxyribonuclease, which is involved in making new strands of DNA. And there's just a variety of different shapes of proteins. There's probably a lot more shape variety available out there than this picture even represents, but some of the main ones that are touched on in a cell biology course are in this picture. These are a couple of images that address how proteins interact with other molecules based on their shape. The four panels to the left in this picture are from the previous chapter in the textbook, looking at how two substrate molecules can be brought together by binding to a very specific part of a protein, and that protein, that enzyme, will catalyze the reaction, bringing those two together, making the product. The other picture, which is from the Encyclopedia Britannica website, shows how the very specific placement of amino acid side chains in a protein can play a role in how it interacts with that substrate. The substrate in the active zone of this enzyme has a very particular shape that fits in to that spot, and it has, for instance, a negative charge or a positive charge in particular places where it can interact with basic or acidic amino acid residues. The four variations on the other side show you how the binding site can be very specific. The first one has a big structure on the side of it, which would not allow that to fit into the active zone, so that's not a substrate. Conversely, the next one shows a gap in that same place, so it's not going to fit into the active zone properly. It'll be loose, if you will. The next one shows how those charges can change. The positive charge in the substrate is negative in this non-substrate, and then it would be repelled by that acidic side chain and not fit in there and be attracted to it. The fourth one is still potentially a substrate here. It has a huge piece sticking off of it, but where that piece is sticking off has nothing to do with the active zone, so it can still fit into the active zone. Now this picture is suggesting the active site of an enzyme, but a good example of this would be a binding site of a receptor. Let's say that what's called the substrate here, called a ligand and a receptor, is a sugar molecule binding to the sweet receptor in your tongue. There are other molecules that fit into the binding site of that receptor and therefore taste sweet, but they're actually different shaped molecules. Those are artificial sweeteners, saccharin, those sorts of things. And it's just a variation in their shape that is the result of not being calorically active, but they can still fit into that space. I said earlier that protein folding is a complex process in the cell, and understanding how proteins fold and how their three-dimensional shape comes to be is a very important part of understanding the proteome, or the complement of proteins in a cell. Computers can do a lot of very useful things to understand what's going on in cells, but protein folding is such a complex task that computers aren't capable of doing it. But human minds are very good at recognizing the subtle patterns that emerge from a process like that. And so this website points to a citizen science website, obviously called Foldit, where anybody can go and sort of play a game where you try to fold a protein. Scientists have taken an actual protein structure and put it into their database, and by going in there you can figure out how it folds. There are certain rules to what can be attracted to or repelled from what, and how things have to work. And if I remember correctly in the website, you sort of get a score as to how well you satisfy as many of the criteria as possible, with presumably the outcome that the best score is going to represent the actual way that a protein is folded. So just having the raw genome data, which we have about all of the DNA in humans, recognizing what parts are proteins, we can then hopefully move on to figure out what shape they have and what function they're going to play based on this sort of citizen science gamification of the process. If you're interested, please go to this website. It's kind of interesting. It might be kind of fun, and you can be part of the process of figuring out how proteins work. Unfortunately, you won't get your name on any publications necessarily, but you can still be a part of it.